Good morning. Today we'll be reading the review paper, one of the review papers on chronobiology. So this one is generation of circadian rhythms in the suprachiasmatic nucleus. So let us start. It is written by Michael H. Hastings, Elizabeth S. Maywood, and Marco Branchi Brancaccio. I'm sorry if I said the name wrong. Okay, so abstract, the suprachiasmatic nucleus (SCN) of the hypothalamus is remarkable, despite numbering only about 10,000 neurons on each side of the third ventricle. The CNS is our principal circadian clock, directing the daily cycles of behavior and physiology that set the tempo of circadian of life. So sorry. When this nucleus is isolated in organotypic culture, its autonomous timing mechanism can persist indefinitely with precision and robustness. The discovery of the cell autonomous transcriptional and post-transcriptional translational feedback loops that drive circadian activity in the SCN provided a powerful exemplar of the genetic specification of complex mammalian behaviors. However, the analysis of circadian timekeeping is moving beyond single cells. Technical and conceptual advances, including intersectional genetics, multi-dimensional imaging, and network theory, are beginning to uncover the circuit level mechanism and emergent properties that make the SCN a uniquely precise and robust clock. However, much remains unknown about the SCN, not least the intrinsic properties of SCN neurons, its circuits topology, and the neuronal computation that these circuits support. Moreover, the convention that the SCN is neuronal clock has been overturned by the discovery of astrocytes are the integral part of the timepiece. As a test bed for examining the relationship between genes, cells, and circuits in sculpting complex behaviors, the SCN continues to offer powerful lessons and provide lessons and opportunities for contemporary neurosciences. So now we'll be starting with the theory portion. Circadian rhythms lasting approximately a day, 24 hours, dominate our lives, most obviously via the sleep-wake cycle. Indeed, almost all vital physiology and metabolism are under circadian control, driven by cellular clocks distributed across the body. These rhythms adjust us to the world by preparing the brain and other tissues to perform very different, often compatible functions appropriate to the anticipated day or the anticipated night. For example, in diurnal species such as humans, neural mechanisms that maintain attention and cognitive capacity are upregulated in the daytime, whereas preparation for the night involves the activation of pathways that are essential for sleep-dependent memory consolidation and reconsolidation and synaptic scaling. Nocturnally active species such as mice exhibit equally robust daily changes, but they are oppositely phased to light and darkness. At cellular level, these rhythms are a product of approximately phased daily programs of tissue-specific gene expression. Disruption of the brain circadian program comprises sleep efficiency and cognitive performance, as well as associated processes such as synaptogenesis and brain metabolite clearance. It is unsurprising, therefore, that disorders of circadian timekeeping have been implicated in various psychiatric, neurological, and metabolic diseases. The nervous system should therefore be seen as the 24-hour machine. But how temporal organization confirmed? There are innumerable cellular clocks across the body, but the principal circadian pacemaker in the mammals is the suprachiasmatic nucleus of the hypothalamus. The SCN is necessary for circadian behavior. It compromises by surgical ablation in animals and by pituitary tumors or vascular diseases in humans disrupts daily rhythms. It is also sufficient for circadian behavior. Grafts of neonatal SCN tissue into SCN ablated rodent store rest activity rhythms. Moreover, the SCN is the autonomous timekeeper capable of expressing circadian cycles of spontaneous action potential firing when it is located in vivo or ex vivo. However, to be effective as a clock, it must be entrained to and thereby be predictive of solar time, and this is achieved by direct retinal innervation. Finally, as the principal pacemaker, the SCN coordinates the subordinate cellular clocks across the body. It achieves this by circadian control of behavioral, neuroendocrine and autonomic glands that entrain cellular clocks in target tissues. The projections from the SCN that mediate this coordination run to local hypothalamus and subcortical brainstem areas. Though through these pathways, the SCN ensures the daily neural and metabolic programs are well defined and tightly intermeshed. In this review, we first consider the molecular basis of cellular timekeeping in the SCN and then consider how circuit level processes determine the emergent properties of the SCN: stability, amplitude, ensemble period, synchrony, and phase disruption that make it such a precise and powerful clock. We focus on a new understanding of circadian electrical transcriptional coupling and the roles of neuropeptideric cues and GABA in directing circuit behavior. This novel understanding will be considered in the light of newly emerging knowledge of SCN circuit topology and neuronal specialization. Finally, we discuss the rodent discovery of the role of astrocytes as the circuit level coordinators of timekeeping. Given the relative simplicity of the SCN circuit and its robust phenotypic properties, it is not unreasonable to aim to understand its principles of action with resolution that has been achieved in some vertebrate circuits. Okay, so now we'll be seeing this figure number 1. It is showing quite a bit of things. So let us see for circadian organization in mammals. A. Let us say A. So this is a part of a brain and it is showing a smaller part of the brain. So let us see what is it showing. The principal circadian clock, the suprachiasmatic nucleus SCN, is located in the hypothalamus, which is highlighted by the box of the coronal MRI scan of human brain. So first of all, this is coronal MRI of human brain, and this part is hypothalamus. The highlighted box of the coronal MRI is located in the hypothalamus. The insert shows an enlarged view with location of the SCN outlined. The the optic chiasm OC lies across the base of the midline third ventricle. This is midline third ventricle. The OC's optic chiasm. This is the ventricle. So here is where it lies across the base of the midline of the third ventricle. Okay, the insert shows the enlarged view of the location of SCN outlined. So this is where the SCN is supposed to be. The SCN okay, now we'll be seeing the B. So this is the B part. This is the B part. Oh god. This is the B part. So we are seeing a brain and the eye and heart and liver and the kidney. So what are we seeing? Different parts of the brain. So here is its SCN in the hypothalamus. It is near the middle of the third ventricle. Let us read out. Okay. The SCN receives direct retinal intervention via the retino-hypothalamic tract. They get the direct retinal intervention via the retino-hypothalamic tract. Retino-hypothalamic tract. Okay. To ensure its synchronization to day and night cycle. Okay. The SCN clock projects to various brain centers, many of which contain local circadian clock that direct behavioral, for example, feeding, fasting, and sleep wakefulness, autonomic and neuroendocrine circadian rhythms. These systemic cues synchronize the local molecular clocks of peripheral tissues. These local clocks in turn direct the local program of circadian genes expression that regulate physiological rhythms critical to the health. For example, rhythms related to the mental alertness, blood pressure, triglyceride metabolism, and renal function. So we'll read it again. What is it saying? The SCN receives direct retinal intervention through the retino-hypothalamic tract to ensure its synchronization to the day-night cycle. The SCN clock projects to various brain centers. It projects to various brain centers, many of which contain local circadian clocks that direct behavioral, autonomic, neuroendocrine circadian rhythms. Okay, these system cues synchronize the local molecular clock to peripheral tissues, and these local clocks in turn direct local programs of circadian gene expression that regulate physiology rhythms critical to health. For example, rhythms relating to the mental alertness, blood pressure, triglyceride metabolism, and renal function. Okay, so through the retinal hypothalamic tract accommodates the SCN to the day and night cycle. Okay, So this now activates whether it should sleep or be awake. Okay, it controls our appetite, it controls our autonomic and endocrine. What happens to the endocrine and neurocrine and autonomic? They control the hormones, temperature and feeding or fasting. Okay, they control our heart, the liver and the kidneys, it is showing. So what in heart? The blood pressure. In liver, blood glucose and triglycerides, digestion, metabolism. Okay, and in kidneys they control the xenobiotic clearance. I don't know what that is. They have local clock. These hormones affect the local clock, and these local clocks affect the rhythmic circadian outputs. Okay, SCN go tells the local clocks. Local clocks go tell the rhythmic circadian outputs. Okay, this is a systematic cue. Okay, these are the circadian behavioral and circadian behavior and physiology. These are the SCN targets. SCN targets them, and then the local circadian clock tells the systemic cues, and they go and tell these tissues local clocks, and then these tissues local clocks activate the rhythmic circadian outputs, which affect the circadian behavior and physiology via blood pressure, blood glucose and triglyceride metabolism and xenobiotic clearances. Another way, the local brain clocks is there. It is also a local clock, but it is a local brain clock. Okay, it affects the brain homeostasis. This portion of the brain also has a local clock which affects the brain homeostasis, and this portion of the brain's local clock affects the cognition and the mood, and they tell their rhythmic circadian outputs to behave in that manner. Okay, now we'll see the C portion. So this C portion, we see a flowchart, simplified schematic of the molecular transcriptional and translational feedback loop, okay, of mammalian circadian clock, in which heterodimeric complexes of circadian locomotor output cycle protein caput clock. Complexes of circadian locomotor output protein caput and brain muscle ARNT like 1 BAM1. Proteins act via the enhancer box EVOX. Regulatory sequences to drive daytime expression of period per, and cryptochrome protein cry, which in turn combine to suppress clock BAM1 activity in their own e-boxes. Subsequent degradation of per and cry in circadian night allows the cycle to start again. Degradation. Additional feedback loops incorporating nuclear receptors, REV, ERB and ROR alpha, are also circadian regulated via e-boxes and control BAM1 expression via REV response elements RREs. This stabilizes and enhances the core TTFL. What are TTFLs? We read somewhere. Yes, this transcriptional and translational feedback loop. In turn, these four ancillary loops drive local circadian program of clock control output genes, the first tier of which are also regulated by clock BAL1 per cry rev ERB and ROR alpha via E boxes and RREs. More complex downstream cascades involving post-transcriptional translation and post-translation mechanism further sculpt the cellular circadian transcriptosome and protein. Imagine in a courtesy of A. Imagine is a part of a courtesy of whatever. Okay. So what is happening? We are basically seeing the molecular transcription translational feedback loop. This is known as TTFLs. Okay, this is schematic diagram of TTFLs, transcriptional translational feedback loops of a mammalian circadian clock, in which heterodimeric complexes of circadian locomotor output cell proteins clock. So clock is a protein whose full name is circadian locomotor output cycles protein caput. Okay, so this and brain and muscle ARNT like one BAML 1 protein. These two are proteins. So these proteins acts via enhancer E box. This is an enhancer box. Regulatory sequences to drive daytime expression for period. So this is this. So we are going like this. So daytime expression of period, and other is cryptochrome protein. This is a cryptochrome protein. Which in turn combine to suppress clock BAML1 activity. So this will work in a negative response, and it will say to stop the BAM1 and cry activity. They will suppress the BAM1 and cry clock proteins. Okay, now after this, subsequent degradation will occur because no more is made, no more this is made, no more this is made. That means this is having a subsequent degradation. That means this negative feedback loop will not work. That means there will be again this BAM1 and clock proteins will be made. They will start again. There is an additional loop incorporating the nuclear receptors rev ERB and ROR alpha are also in rev ERB and ROR alpha are also circadian regulated via E boxes and control BAM1 expressions. BAM not, it's BAML. sorry, BMAL, BMAL, BAML, BMAL. Okay, BMAL one expression via rev response elements. So rev response elements, RERs, rev response elements. Control this controls BMAL expressions. This stabilizes the enhances the core TTFLs, transcriptional translational feedback loops. Okay, in turn these core and ancillary loops drive local circadian programs of clock control output genes. So listen, this ROR alpha makes rev regulatory response elements. This rev, same thing. Okay, and this one inhibits rev ERB, inhibits RRE's formation. Okay, so this one is inhibitory, and this is what is shown. RREs enhances the BMAL1's production, it enhances its production. Okay, after this we see it in this stabilizes the core TTFL, transcription translational feedback loop. In turn these core and ancillary loops derive local circadian programs of clock controlled output genes, the first tier of which are regulated by clock BMAL1 per cry and rev ERB and ROR alpha via E boxes and RREs. More complex downstream cascades involve post-transcriptional, translational and post-translational mechanism further sculpt the cellular circadian transcriptome and proteasome. Okay, understood? I think so. It is simply saying there are these two proteins, okay, BMAL1 and clock. These are the proteins. They work through they go through the E boxes, which is an enhancer. Okay, and this gives a circadian output gene. Okay, so they also make this per gene and cry gene. The per gene is daytime expression of period and cry is the cryptochrome protein, but they give a negative feedback loop and they make the E box to stop transferring and whatever, stop the they make the E box to suppress the activity. They were enhanced, it was an enhancer, right? So it will suppress now the activity of BMAL1 and clock proteins. But now that it is not anymore enhancing, it is suppressing, so this is not being made anymore, this is not getting enhanced, and no more per and cry are being made. Okay, so this is this makes it goes to a subsequent degradation of per and cry, and circadian nights allow the cycle to start again. So this cycle will start again. So other than these two, it also forms rev ERB and ROR alpha. So it is seen that they affect the RER, rev response elements. Okay, so the rev ERB suppresses the RERs and ROR alpha enhances the RERs. The RER is what is important for the expression of BMAL1 protein. Okay, This cycle enhances and stabilizes the transcriptional translational feedback loop. That is what is said here. Okay. Now the molecular clock. The award of 2017 Nobel Prize Physiology or Medicine to Jeffrey C. Hall, Michael Rosbach and Michael W. Young for their discoveries of molecular mechanisms controlling the circadian rhythm is a testament to the fundamental importance of circadian clocks. However, the award also does something else. It highlights the achievement of deciphering how complex behavior is regulated by genetic and molecular mechanisms. Work by these leucotrienses in the fruit flies and its translation by many others of higher species has revealed the elegant and sophisticated nature of circadian timekeeping in mammalian tissues and cells. The optimistic mantra genes to cells to behavior is often chanted at the start of neuroscience seminar, but with clocks we really do have that spectrum to understand. Cellular circadian timekeeping in the SCN and other tissues pivots around self-sustaining transcriptional translation feedback loops in which beginning at circadian dawn started at the neuroscience seminar, but with clocks we really do have the spectrum of understanding. Cellular circadian timekeeping in the SCN and other tissue pivots around self-sustaining transcriptional translational feedback loops TTFLs in which beginning at circadian dawn, that is circadian time TC0, heterodimers of circadian locomotor output cycles protein capotes clock and brain muscle ARNT like 1 BMAL1, also known as ARNTL. The positive regulators of the loop drive the expression of period PER and cryptochrome cry protein. The negative regulators via the enhancer box E-box regulatory sequence seen in the figure. By the end of the circadian day, CT12 per cry complexes have accumulated in the nucleus and start to repress their own expression. Therefore, over the course of ensuring the circadian night, CT12, CT24, CT0 per and cry mRNA levels fall. The existing per cry complexes are degraded. This degradation allows the cycle to reinitiate approximately 24 solar hours after the previous transcriptional initiation. The cycle is stabilized by the accessory loops in which clock and BMAL1 drive E-box mediated circadian expression of the nuclear receptors ROR alpha, REV, ERB alpha, also known as NR1D1 and REV, RER beta, also known as NR1D2, which in turn acts via rev response elements RRE sequence to activate and suppress BMAL1 transcription respectively. Mutations in these genes of these nuclear receptors alter the amplitude and period of activity rhythms. The Rev ERP double knockout mice exhibit severely disrupt circadian behavior. Moreover, administration of the synthetic Rev ERB agonist can suppress the amplitude of SCN TTFL and nocturnal behavior in the mouse. Thus, an output of the core TTFL circadian expression of Rev ERB protein is also an input to it. The molecular mechanism that support the TTFL are now being explored using mutational, biophysical, and structural biology based approaches. Supplementary box 1. Major advances have been made in understanding how the positive and negative transcriptional regulators interact at the molecular level over the circadian cycle, and how changes in key residues and motifs affect these interactions to alter transcription and stability of per cry complexes and thus circadian timing. The time cues defined by changing molecular status of these interlocked feedback loops are transmitted to the rest of the cell to coordinate its activities. This is achieved by circadian output genes, the first tier of which also incorporate EBOX and RRE sequences, and thus sequentially upregulated and downregulated over the course of circadian day and night as levels of per cry, rev ERB, and ROR alpha wax and wane. Many output gene encodes transcription factors, allowing the core loops to temporarily coordinate the activities of hierarchical cascades of the cell type specific genes expression that ultimately guide the cell functions. So I'll take a break here and we'll start from this SCN neurons as circadian pacemakers. Okay, thank you, bye bye.